In this video, we will learn about Ames test for the detection of potential mutagens.
As we know, chemical carcinogens can induce mutation which can lead to tumorous or cancerous growth. And normally we test for mutagens or carcinogens on animals. But animal testing for all compounds will turn out to be a lot expensive and time consuming as well. Thus, we use Ames test. The benefit of using Ames test is that it is a really simple process, it is inexpensive, and it is an indirect assay for testing of mutagens. Consider this is a molecule which need to be tested for its mutagenic or carcinogenic activity. And after testing, you come to know that this compound is not mutagenic. In most of the condition, you will go ahead and use this compound in different applications since you are assuming that the compound is not mutagenic. But consider the same compound after getting metabolized in your body. Assume that this compound was in a food product and that food product you've consumed. After consumption, the compound is going inside your body and it is getting metabolized into small products. Now, these metabolic products that are obtained, you are collecting these products and you are testing them for their mutagenic activity. And you come to know that these products are mutagenic. So here, the actual compound is not mutagenic, whereas after getting metabolized, the products that you are obtaining, those are actually mutagen. So these products are mutagenic. So you need an assay that can test for the metabolic products as well as for the compound as well. In Ames test, we use Salmonella Typhimurium as the test culture. We mainly need to check for the mutagenic potential of the metabolic products. And for that, we want our test compound to get metabolized. Since Salmonella Typhimurium is a bacterium, it cannot synthesize the enzymes that are required for the metabolism of the test compound. Therefore, liver enzymes are added so as to carry out the metabolism of the test compound to get the metabolic products. Before we move on to the actual test, let's clear some important terms. Prototrophs or wild type are organisms that have the ability to synthesize all the compounds that they are needing for their growth. These organisms are self-sufficient and they do not rely on the essential compounds to be supplied in the medium. They can synthesize their own compound and they can grow in the medium. Now the second term is about auxotrophs or mutants. These organisms, they lack the ability to synthesize either one or more essential compounds. So if you want to grow an auxotroph or the mutant, you must supply the particular compound in the medium so that the organism can utilize that compound and it can grow. Now revertants are organisms that can revert back to the wild type, or in simple language I can say they are auxotrophs or mutant that are mutating again to go back to the wild type or the prototroph form. In this experiment, we use Salmonella Typhimurium culture, which is an auxotroph for histidine, which means that it cannot synthesize histidine on its own, and to grow this organism you need to provide histidine in the medium. Another thing that you add is rat liver enzyme so that the test organism can be metabolized into the metabolic products. In one of the plate, that is the test plate, you add the possible mutagen and you incubate this plate. Now this plate contains very minimum amount of histidine. You can consider that this plate lacks histidine. So the organism, which is a histidine auxotroph, will not normally grow on this plate. it cannot grow on this plate. But only if this organism is mutating due to the mutagen, there is any kind of mutation happening in the test organism, you will get colonies on the plate, which will indicate that the strain is getting mutated due to the possible mutagen that you are adding. We also keep a control plate wherein you don't add the test compound. We know that mutation can be random or induced. So, in a condition where you are not adding the possible mutagen, in that condition also there are chances that the test culture will show you some amount of mutation. That's why you keep a control plate so that you can check for the presence of natural revertants. Now, natural revertants are basically mutants which are having a mutation which will change their histidine auxotroph back to the wild type form. So, if the test compound is not mutagenic, the number of colonies on the test plate will be equal to the number of colonies on the control plate. Not equal, but it will be approximately the same. But if the test compound is mutagenic, it will induce a large amount of mutation in the test organism, that is in the Salmonella Typhimurium culture, and you will obtain a high number of colonies, which will indicate a really high number of revertants. So, if the number of colonies on the test plate is more compared to the control plate, in that condition you can conclude that the test compound is actually a mutagen. Now consider this first example wherein you are adding the liver extract as well as the test compound. Since the test compound and liver extract both are present, the test compound will get metabolized by the liver enzymes and you will obtain the metabolic products. If the product obtained after metabolism are carcinogenic, you will get a high number of colonies, that is a high number of revertants will be obtained. Now in the second condition, you add the liver extract but you don't add the test compound. Since the test compound is not present, all the colonies that you will see on the plate will be your natural revertants. Thus, this plate will act as a control plate for your experiment, which will give you a count of all the natural revertants. Now, in the third example, you are not adding the liver extract, but you are adding the test compound. In this condition, the test compound cannot get metabolized as the liver extract enzymes are not there. In this condition, if you get a high number of colonies, then you can say that the test compound was also mutagenic or carcinogenic. But if high number of colonies are not obtained in this condition, but you are getting high number of colonies in this condition where you are adding the test compound with the liver extract and not in this condition, then you can conclude that the test compound was not mutagenic, but upon metabolism, the metabolic products of the test compound are actually mutagenic or carcinogenic. Like any other test, Ames test also have some limitations, like it may show some false negative result, that is a compound may be mutagenic upon metabolism, but on Ames test you conclude that the compound is safe. Also, animal tests are required to confirm the results that are obtained from Ames test, because Ames test is not a really reliable method.